Danke kindly, Dominik. Danke schön. Ich freue mich wirklich, mit euch zu sprechen. Das sind Themen, die ich so gerne darüber spreche für Audiences, die zwischen diese beiden Kanten sind. No? So I already see here, and I'm delighted to notice that there's a set of multimodal distributions here, and really bipolar, bimodal distributions in many of these essential topics, which amuses me because I've been living precisely in this middle point where no one is, so the sort of this rarefied stratum between the two extremes of artificial neural networks and biological neural networks. In the Erasmus now, I'm assistant professor for computational neuroscience. I am currently, I have a tenure, and so what we're going to be doing forward going is building on more of a catalysis between the biological sides of everything and the synthetic sides. I also work for the TU Delft currently, so I don't know if you guys are familiar, but it's a good engineering school here, and I teach in the nanobiology program, where they are also doing this sort of trying to, in this multidisciplinary world of ours, to stay comfortably in the middle. This is the kind of stuff that I've done before. Actually, the work that I've done in the Fraunhofer was related to the square on the left. And if you cannot see something, or if you want to ask a question, please interrupt me at any given time. Okay, so I've worked on evolutionary robotics, which was the idea that through evolutionary procedures, you could create recurrent neural networks that controlled the robots, and that was the work that became a PhD thesis in the Fraunhofer. And there I was studying the dynamics of recurrent neural networks more than simply categorization. We were looking into dynamical systems perspectives, and I want to look into the results of the poll in a moment to discuss it. But having studied the dynamics on recurrent neural networks, which are relatively artificial, I became convinced that there were analogies between the synthetic world and the biological world that were very compelling. So I was impressed the degree of which these simplified neural networks could get us insight into the function of more complex brain parts. And in fact, this is now what occupies me here in the middle bottom corner. It's the anatomy and dynamics of the cerebellum. So these are also recurrent neural networks, and in many cases they have similar function. The only problem is that they're much more complex than the kinds of small neural networks that I was dealing with when we were working with evolutionary robotics. And we don't know what they're good for. So in evolutionary robotics, one of the things that we do is we have a guideline, which is we know what the fitness function is. We know what we want to achieve, and so we create that as to permit our gradient descent. Whereas in the case of the brain, often we don't actually have all of the access to the objective functions of the fitness. So we have to do a lot of reverse engineering, and that's what we're doing when we're modeling individual cells at the level of physiology. So what you see here in the middle is an inferior olive of the cerebellar system, which is supposed to be partly responsible for the control of motor behavior in the brain, only that we don't really know how exactly that happens, even though there have been assumptions about and analogies with artificial neural networks. So this is just a little introductory slide. I'm shamelessly going to tell you that I wrote a book that I still think has some of these ideas, captures some of these ideas, and I still sort of stand by the kinds of stuff that I wrote there, even though it was like 8-9 years ago during my PhD, as to how to approach the problem of reverse engineering the brain. What, in fact, are the invariants of behavior? But I'm going to get to that during our presentation as well. So before I go into the next slide, I wanted to, so can you still see what I'm seeing? So I wanted to discuss what I see here. So it appears that in the crowd that is watching, you have, like I mentioned earlier, a bimodal distribution in the level of confidence from biological neurons, for instance. So you have, I have in the audience some of you know quite a bit of it, and some of you feel a little bit more shaky. So I imagine that there is some sort of correlation between the left extreme here, in which you're basically both rating your knowledge of biological neural systems, and also then the artificial neural systems. So there's an interesting split. But dynamical system seems to unify both sides, or at least I appreciate noticing that there is a peak in the dynamical system that brings the two that conjoins the two extremes, even though still polarized. Because dynamical systems is a great language for us to talk about the brain. And as I mentioned a moment ago, I make neural models. And this, for instance, is a neural model. This is a fractal model that shows you a two-dimensional neuron that is created by a context-free grammar that is written on the left. So I wrote this piece of code in this piece of software called context-free, which is a stochastic fractal generating piece of software. And I often start to talk by when I'm talking about modeling the brain with the discussion of what is a good model. I can sometimes convince, actually fairly often, I can convince even biologists that these neurons that I produce artificially and that live in a 2D space with growth rules that are simply, branching and producing more of the same. So just transformations, visual transformations. I can sometimes convince biologists that these are real neurons. And they ask me, where's this neuron from? And then I asked them, what is a good model? So what is a good model of a neuron? This can, it has surface features of a neuron, but it does not necessarily has explanatory features with respect to brain function. It's mostly a visual, it is a good visual model, but it's not a fundamental model. Which brings me to the next question here that I wanted to share in this presentation, which is a poll that we're going to come back to later. So if you already have the Mentimeter open, then you can come to this slide. And yes, I wanted to probe your brains again. Now you've rated your brain, now you're going to get a mirror and try to see, well, is it an understandable problem? I don't know how many people we have inside, but this is very common, what we're seeing here. This 50-50 is actually generally, nice, okay, so the straddlers now are coming. Still having some sort of representation of, it seems unbalanced. But still, I mean, there are some people that actually think that we can understand the brain. However, this question, I think, is only meaningful if we are able to interpret, to produce a definition of what we mean by understanding the brain. And hence, my next slide. I'm going to assume that you are 18 people, and I want to see how you change your minds as we go to this one instead. What is understanding the brain? But really, can we understand the brain? So you guys that said no, I presume that partly you're thinking, Well, I mean, no, but there are parts that we can actually, get our heads around. And the question is, what is the part of the brain that we actually can understand, and how will it satisfy us as onlookers that we're actually understanding the brain? And there is a number of ways in which I would argue that we can understand the brain in very profound ways, in fact, very mechanistic with respect to cognitive function, the kinds of things that the brains do. So this is then, I think, a more representative split between the minds, because ultimately what we want to really do when we're saying we want to represent the mind is to give the caveats, right? I like to have these as basically discussions, but because I have quite a bit of material to present and we're already 15 minutes in, I think I'm going to try to speak for you, as I imagine that when you say no, but you're thinking, well, there are many things that we can break apart and understand. And I would like to propose that the no, but is, or the yes, the yes, but perfectly splits, they represent about 90% of the brain. So I would say that, the actual function that the brain performs so that we could embed in a robot and make it compellingly so that it looks like us would be a very good inlay towards understanding. We might not be able to finally manage the qualia question and the consciousness question, which I think it's also one of these watersheds. But we will be able to mechanistically understand how information processing, looking at it as systems, as information processing devices, how the influx of information mixed into the anatomy of the brain can then produce the behavior that we actually want to explain. And that's the main explanandum of the brain. So thank you for this 50-50. I've been applying this slide in a couple of different contexts. And in technical contexts, I usually get a balanced version of these two slides. Can we, or but really can we? 50-50 seems to be a very good representation, which I think actually brings us indicates to us that we think relatively similarly. So now, because Keynote is, okay. And so this is a model of a neuron, but it's not a very good model of a neuron, or maybe it is a good model of a neuron. And the question is, what is a good model of a neuron in the end? And please interrupt me with audio if you want to ask questions, because I have, so given my setup here with the screens, it is hard for me to tell that you're actually saying something in the chat. I don't even have access to the chat. So basically just jump in with hello, and I'll address your question. So I wanted to try to understand what is it that we can understand in the brain. We can have different approaches. And I feel that computational neuroscience and neural computation present complementary but distinct ways to look into this question. And when we're modeling the brain, so as the first question we just discussed, which we haven't discussed yet, is what is the language that we should use? Because when we're modeling something, when we're trying to understand something, we're usually trying to create explanations to ourselves. We are trying to use language, mathematics, or English, or to try to wrap our heads around what is the language that we should use when we're modeling the brain. So we explain, okay, well, that person made that decision because that person believed that thing in that context would obtain, and so she took the this course of action. So this is called what I just this abstract representation of a decision-making procedure is what people in philosophy call the theory of mind, the idea that there is a reason for us to act in a certain way, and this is also being called folk psychology. It's one of the languages we sometimes use. And so models are representations of our beliefs, and what is a good model is something that tries to get to the core of the explanation. So a good model is then representative, explanatory, it's consistent with other known models, at least to the extent that it's talking about the same thing, is predictive, quantifiable, but encompasses the phenomenon also qualitatively. It's good when a model is simple because you can understand it, but it's also good that it's detailed because then you can see what matters. It's good when it's accurate because then you can predict precisely, but it's also good when it's understandable. One thing that should become clear from this slide in the torrent of my speech here is that many of these goals for modeling are contradictory. I mean, it's quite hard to have a model that is both simple and accurate at the same time, for instance. Or it's also often hard to have a model that is both predictive and explanatory. When people are talking about complex systems, so the theoreticians that talk about complex systems often think that in the context of complex systems like the weather, a simple model is not necessarily the best model, because often what you're looking for in understanding a complex system are qualitative statements about what the system looks like. So the weather, you can predict the weather in different ways. You can try to understand the weather and you can try to predict the weather. These are not necessarily the same kinds of models. And to cast a light within this space, to try to understand what level of analysis we should take, different people will take different levels of analysis. I made once a mind map of the disciplines that were related to try to understand the brain computationally. This is then something about brain science here in the middle. I'm actually bring you to my newest version of this mind map, which is here, and start from, I forget here what's my what's my shortcut to collapse. But these would be, to my mind, some of the 5 fundamental branches that allow us to tackle questions about the brain. I won't go very detailed on this, but it's clear that at one point we have the biology of this, which is the neurobiology. So we found out that there are neurons. You know, Santiago Ramon y Cajal, 120 years, 30 years ago, found out that neurons are individual things. There's not like the circulatory system. And then we found a host of other properties of neurons. It has a membrane, and like that membrane produces electricity across the membrane. We can measure what this electricity looks like. We can make experiments. So the neurobiology of things will allow us to create experiments. On the other side, we have the psychology of it all, which is the so-called cognitive neurosciences, which are looking at the brain as an apparatus that produces cognition. Cognition being the set, so different definitions have been proposed. I mean, I will just shoot a working definition here, which is cognition is the set of functions that the brain affords to an organism that allows it to interact meaningfully with its environment. And I underscore the word meaning here because when we're dealing with the environment, we're dealing with things that represent functions, like affordances, if you allow this vocabulary, that represent things that we can do, and that's like very meaningful patterns. You know, what is an apple, for example. And so this will include both the input systems, like in perception, and they will also look into cognition. And these are some cognitive functions. The mind map is not supposed to be precise. It's supposed to, bring up these ideas. So pattern recognition, attention, language, symbolic reasoning, creating predictions, having some memory that both of gestures and motor behavior, or also episodic. You can remember something that you saw in the past, navigating to a different place, imagine something, produce a decision. So this is what we want to explain when we want to explain the brain, at least to my mind. So this is the thing that brings neuroscientists into neuroscience. It's like, okay, so how does the brain work? And so this is the stuff that it does. And this is what it does it with. So the brain does cognition via the neuron. And To start our research, we look into theoretical aspects of neuronal functioning, and we notice then that it's a very dynamic system, in the sense that we seem to have neurons with physical properties that imply certain specific types of dynamics. And on another hand, we can also quantify the statistics of neuronal activity, neuronal coding, and to look into features that are being encoded and decoded in these receptive fields and so forth and so on. Computational neuroscience is the effort of simulating neurons to try to get at brain function. Only that it cares a lot about the accurate representation of the biological systems. It looks at the biologicals, it thinks it's a bottom-up approach. It looks into models that are fairly detailed and tries to analyze the biological, biochemical, electrobiochemical components of the neuronal tissues, such as the brain. Whereas AI, and I put here like blatantly under the umbrella of neural computation and cognitive modeling, is trying to get to assemble that function that we observe from cognition. So if on the one hand we take neurobiology and simulate it by creating via computational neuroscience, neural computation is trying to reproduce artificial neural networks via things that look like the brain with strong simplification, certain functions that are analogous or attempt to be analogous to those that are produced in our brains, such as learning, associative learning, and then we have other components here, such as supervised learning or reward learning, unsupervised, and so forth and so on. So present to you what to me seem like complementary ways to look into this. And when we're asking the question, can we understand the brain, is what does it mean to understand? It really means to answer like part of these questions. Can we understand, for instance, how a neuron produces an action potential? Can we understand how a neural network produces pattern recognition? Can we, or a brain neural network produces pattern recognition? Can we understand how the local field potentials, the electrical potentials in the tissue for electroencephalograms, EEGs, are generated in the brain? That is the core of the understanding question. And that's what the talk is about. Naturally, we start with neurons. And this is a lovely simplified picture of a neuron from one of the standard encyclopedias of neuroscience, in which you see exactly that, what everybody imagines. A neuron isolated in space, it receives its information from its apical dendrites on top, integrates that in the cell body, and sends the signals to receiving neurons via its axon that distribute then information. And while this is a very useful idealization of what a neuron is, I propose that it's also very important to remember what these guys actually look like in biology. Even if we see them as a little isolated components, these cells here in yellow, there's just few. It feels like they're kind of floating in space, but more and more we get amazing pictures of this very thick wiring that looks like that really embodies what we now believe in neuroscience. These are not the neural networks that we do often. They don't look very much like the neural networks that we do in artificial intelligence, because they're packed. They come originally evolutionarily from skin. So the neuron is an evolutionary sophistication of skin, and you will have many short connections, very packed short connections, topographically organized, not only topologically organized as in artificial intelligence. And we also know that they express a whole host of different kinds of proteins. This is a rainbow picture, which is a method to look into individual neurons according to their biochemical status inside of them. So each color here represents a neuron that can have different genes being expressed or being in a different activity state. So we see this incredible parameter multiplicity, incredible biological variability at that level. And neurons produce spikes, which are the action potentials, the electrical potentials that are then integrated in the soma, conveyed to the axons. The standard idealization is through synapses, or synapses are basically little current injections into the neuron. And when the current injection is sufficient, you get an all or none potential. So that's the idealization. Better than the I have a slide missing here. We're going to come back to this slide. But so the idealization that we have is actually capturing only a very small proportion of what neurons actually do. We have spontaneously firing neurons, we have bursting firing neurons, we have neurons that don't need any input to fire, neurons that only fire when you inhibit them, which is the opposite of what you might expect. And there are a lot of these sophisticated parameter space analysis that you can conduct via neurodynamics, which is one of the topics of computational neuroscience. So when we look here into these fundamental differences, what we should focus on is the disambiguation between these two extremes. Like one, the idealized functional view of neurons, and the other, the experimentally drawn physical view of neurons. And I guess that you already got my point, but to belabor it, I was just going to disambiguate it via this matrix. In computational neuroscience, we have a low level of abstraction. That means that we are looking at things in exquisite detail. A low level of abstraction means you're not abstracting very much. You're trying to get to that what you actually have, ion channels, the actual membrane. So the focus then is an experiment. You know, how much calcium is in the cell, how much DNA. You measure it a lot. What's the amplitude of the potential? You create a hypothesis test of the tissues. So you ask, okay, what happens if I get this type of an input of this amplitude? It looks into biophysics, really the physics of the neurons. When it looks into the changes of the circuit, it talks about the changes of the circuit as plasticity of neurons. When it talks about how the neurons are wired, it talks about anatomy. Neural computation, on the other hand, will talk not about plasticity, but about learning, the one thing that has been acquired, the thing that the circuit now is able to do. It learns, it acquires a function. No, learning is not simply changing, it's changing in a good direction, or changing in a memorable direction, if you like, in a well-organized, meaningful direction. And instead of talking about anatomy, we talk about the circuit. So what are the ideas that evolution was trying to get in to solve the cognitive problem? So in neural computation, we're really focusing on behavioral function. We're focusing on explanation for function, or engineering something. So that's why our language is more like computation in neural computation, whereas in computational neuroscience, our language is physics and neurodynamics. So I guess the emphasis is computational neuroscience, the emphasis is neuroscience, and neural computation, the emphasis in computation. The bottom line, hence, is that with computational neuroscience, we simulate neurons, and like in the Human Brain Project, I'm going to give you some examples of that. And in the neural computation, we compute with neurons. So here's how computational neuroscience looks like. It looks like a fully reconstructed Purkinje cell of the cerebellum, where we have substituted the pieces of the dendrites that we've measured from a microscope to little cylinders that have bioelectrical properties and ion channels. This is how neural computation looks like. You have numbers coming in, a bubble, and the bubble sums those numbers, applies a saturating nonlinearity or other kinds of nonlinearities, and produces another number which it distributes to other guys. Let me focus first on computational neuroscience. The onset, so the dawn of computational neuroscience was the idea that you could model the generation of the action potential via an electrical circuit by Hodgkin and Huxley, merited the Nobel Prize winner. So this, the Nobel Prize, this is not 1990, of course, this is a reprint. No, I mean, the original paper, I think here is from 1952. So the main idea, well, I guess that many of you would be able to parse this, but ultimately it's a very simple circuit where you have variable conductance of two types of ions, sodium and potassium, and you have a leak type static constant conductance that goes that brings ions to and in and out of the membrane. The generation of potentials in the membrane is due to the difference of ion channel concentrations inside and outside of the cell. So you see that the level that we're actually discussing the neuron and the action potential is definitely not digital, it's very physical. We are trying to understand it as a function of the ions that are inside and outside the cell. And it produces spiking neurons. This is the this is the Hodgkin and Huxley, famous Hodgkin and Huxley equation. Not exactly the same, so you don't need to understand the entire thing here, but I wanted to tell you that basically this is current in equals to current out. And you have these different components, I, K, I, and A, and I leak. But ultimately, this is a dynamical system of four variables. The V dot represents an ordinary differential equation where we talk about the difference of the change, the rate of change of membrane potential over time, V dot. Then N dot, M dot, and H dot. These are the gating variables. The gating variables tell you how many ions are crossing through the membrane. They are numbers that go from 0 to 1. And so here we have a dynamical system, a four-dimensional dynamical system that allows us to understand the production of spikes. And we can look at this from the neurodynamics perspective and look into the bifurcation diagrams and so forth. And so we understand the neuron and the production of action potentials as an interplay of the state variables of the dynamical system here, the gating variables, which have their computed solutions, their simulated solutions. These are forward Euler solvers. That's where, you know, my friend Christos next week is going to talk more about the computational solutions that allow us to simulate effectively these kinds of processes, because ultimately, yeah, one of the main questions is, you know, how to solve this differential equation. It's a fundamental problem that leads us to our computational caps. The simulation of our neural network models have to do with this solution of this equation, which is represented by these four differential equations. It's time on the bottom. I am not explaining the graphs like fully, but I'm happy to discuss them with you later at any given point. And as I was saying before, like, these are action potentials. The action potentials are heterogeneous. They're very different from different cells, but we have figured out ways to simplify the Hodgkin-Huxley model via mathematical considerations and neurodynamics type models. And you may have heard about the Izhikevich model, which is a two-dimensional model, not a four-dimensional, but a two-dimensional model that can be parameterized to produce input-output functions that look like most of the classes of neuronal firing encountered in real life. So if now I tell you that the neurons are not simply ons or offs or zeros and ones, and I tell you that there's a lot of analog type encoding here, You should be looking at these 16 different types of neurons, and they're basically responses to input currents. So there's an input current trace here at the bottom, and there is a membrane potential, action potential on the top. These are 16 different types of neurons, and they have 16 different types of functional properties, of computational properties that will be used then for from the brain to get stuff done, to get cognitive stuff done. Only that we don't really exactly know how to do this yet. What we do know is how to produce action potentials that look very well and very compellingly like the action potentials that we see in real life. Now, I just wanted to show you this because I think it's so cool that one of the first two-dimensional neural models looked like a radio. This is the Fitzhugh-Nagumo model, implementing two differential equations that have been solved by this circuit here, right? So ultimately, analog encoding. I think it's amusing to think about. So we have, from one hand, the relatively complex multi-compartmental model of the neuron like this one, where we are looking into very complex ion channel state variables. And on the other, we have much simpler models that are simply producing spikes or membrane potential given a certain current, that are more computationally efficient, but perhaps not as accurate. And that's an interesting thing. Again, when we're talking about explanation, what is it that we're interested in? Are we interested in reproducing the types of spikes, or are we interested in reproducing the exact firing of a neuron? Here we have the abstraction of a compartmental model. So the Hodgkin-Huxley was a single compartment model. It imagined that the entire neuron was a ball. And so the axon of the squid, which was the experimental paradigm that it had, was actually a ball, and that ball had a capacitance and it included some ions inside. But we evolved to think that neurons are like these compartmental versions of themselves. They're not simply bubbles, and the different compartments may have different combinations of conductances, not only potassium, sodium, but maybe chloride, maybe calcium, many other different kinds. And we model them via equations like this, in which we have a passive leak from across the compartments, and we have like some active mechanisms that are creating the voltage differences within the compartments. Building from here is the Human Brain Project. So this is the coronation of the Hodgkin-Huxley model. And you can see here that nowadays computational neuroscience often has cosmology-like author lists, because it really is a huge amount of work to produce accurate representations of the neurons in the brain by tracing them, for example, and by categorizing the vast variety of different neurons in the brain to see how they fire. Here we have the representation of neurons in this plot. We have a representation of the structures of neurons from the Human Brain Project in blue. We have dendrites, and in red we have axons. These are different kinds of cells. They coexist in the cortex. And people have said, well, you can't do this, man. Like, it's way too complex. You're starting too fast. You don't know enough. Well, I think that there are good arguments to say that we are at the point that we should really try. And I'll try to convince you of that. So this is a reconstruction of a single column by the Blue Brain Project prior to the Human Brain Project by Markram, in which they've reconstructed an entire column of neuron by neuron of the mouse cortex with all known cell types. And so these are the cell types being highlighted as you are rotating in this three-dimensional representation of the column. These are the synapses on this neuron. So it basically receives synapses on the dendrites, and it tries to receive synapses in a way that are accurate somehow. So actually, this is the activity of the column, but allow me to move forward because we are at 10:44, and it would be nice if we had a little bit of a discussion. So we can then reproduce the activity of that entire column and look, well, does it look like reality at all? And it is very compelling to see. So I'm not going to explain this entire slide, but A shows the neurons, B in their cortical layers, B shows the spikes of the different neuronal categories across time, C shows the spikes that come from a certain input, and D shows the overall histogram count of the spikes from the input. And the funny thing is that even though we add all of this complexity and everything could happen, not everything happens. What happens are things that are closely related to how we've reconstructed the neurons with all their physiology. For all of these different neuronal categories, we found the parameters that brought them to be more or less what they are in reality. And when we simulate this tissue, it really looks like tissue, right? So like the organic tissue that the brain looks like, very packed, very dense. We see statistical properties that bring us back to the original observations. So here, is going to go. So this is a neuron that or a reconstructed slice where you see here activity propagating from time to time and a little bit of a seeming oscillation. Neurons go from blue, which is hyperpolarized, to red and yellow, which is depolarized, and this is simulating an in vitro slice of brain. And so we see some of these propagating waves and so forth and so on. One of the things that turns out is that, I'm actually advancing the movie here, is that when you change the parameters to represent not the in vitro situation where we measure the neurons, we produce a virtual slice that is in vivo-like, most of the neurons are way more active for the majority of the time. And it looks, in terms of statistics of firing, very compellingly close to what we actually observe. So it does not have those slow wave propagations that we saw in slice. An experimentalist, a neuroscientist experimentalist, might actually, take neurons from the brain, put them in a cold slice, try to measure them, and he will find them oscillating, where in reality they probably wouldn't be oscillating. In fact, now this is not only a prediction from the Human Brain Project, but also a compelling result. It explains how the physics of in vitro slices are different from in vivo slices. And these are 18 predictions from that paper. Predictions. So, the good science produces predictions. And we're going to go into here and discuss each single one with the experimentalist in the context of the neuron. I just wanted to give you a view of what are the goals of computational neuroscience. And one of them is to produce predictions via these highly complex simulated systems. But we can go much simpler. We can go way simpler. This is a very old paper, but I keep it here because it has this simplification thread from the detailed compartmental models to reduced models to single compartmental models like Hodgkin-Huxley, to these, you can already recognize here the non-linearities. I mean, so these would be single population models. I mean, like stuff that's closer to neural computation, all the way until a black box model. And you see here a representation of probabilities. Given a probability of stimulus, you have a probability of a response. Finish that. Take these simplified neurons and try to see how they compute. Neural computation is the idea that we're doing stuff with the neurons. So pattern cognition, categorization, object tracking, motion perception, decision making, motor control, navigation, pattern generation. They are all examples of things we would like our computers to do the way that our brain does them. But we shouldn't forget that the brain receives patterns as input. So it receives highly patterned input from every single sense organ. So on the left here we have the source of physical perturbation. In the middle we have the sensory organ, and on the right side we have the modality that we get from that particular pattern. But all of the leftmost physical properties can only be interpreted by us when they're producing meaningful statistical regularities via this sort of transduction. The idea that we're converting energy into brain energy. So the spikes are going to be the one thing that is going to bring patterns back. You might have seen something along these lines. Modern neural networks, this is from 2016, so now we have different networks already, but it is about putting these circuits together to do patterns, spatial temporal pattern stuff. Not only spatial temporal, sometimes simply spatial, but ultimately, things like echo state networks. Here, I don't know if you can see my cursor, but on the bottom, the second to last line, this is the invention by Herbert Jäger from the Fraunhofer to produce spatial temporal patterns, which is an absolutely genius idea. And in any case, most of these networks use very, very similar units. And their function is different because their shape is different, ultimately. But as we make them more complex and we make these networks reproduce biological function, they become more and more complex, like a convolutional neural network here and on the right side. This is just a visualization of a modern deep learning network where you get these convolutional layers, and guys can already probably, if you came from neuroscience, you would look like something like this convolution 2x group here, say close to the center at about 8 o'clock. If this would be a clock, you can see that it has this ganglion-looking aspect, and many of them have it because of the convolutions. In others, you have convergence and so forth and so on. So these circuits become more and more similar to what aspects that we actually see in biology. So we become more and more compelled that we're not going very wrong. And I just wanted to give you a notion that so we find these invariances when we measure the brain. We encounter certain types of invariances that bring us back to how modern neural networks working today are helping explain certain abilities of the brain. So an invariant of something, right? So like, actually I'm not using the physical definition from Emily Noether. I'm more saying that more general is that what is kept after a transformation. For instance, I can reach a cup in two ways. That means that from the perspective of the function that I'm executing, the function is invariant. Might be completely different networks in my brain, but what I have reached is invariant. So what you want to find is the fact that I've reached something is an invariant, has some sort of essence. But obviously, you have the more trivial invariances like. Identity of an object is invariant with respect to variations of the stimulus. So you would like actually to have invariant recognition in many of these cases. And all of this goes through the idea that in the brain we have cells that respond to features. Here you have on A oriented bars that are presented to the eye, and on B you have the response of a recorded cell in hertz. So that's firing rate of a cell as a function of the orientation angle. So basically this cell likes it when the orientation of the stimulus presented is completely horizontal. So this cell then becomes a pattern recognizer for that particular stimulus. So that's nice. I mean, it basically gives us orientation detection. So this cell is orientation invariant. This is a tuning curve, and you have tuning curves for sensory systems as you have for motor systems. These are monkeys reaching things in 8 different directions. Also with the tuning curve that looks Gaussian. This is a tuning curve, feature extraction from 4 hair cells of the cricket leg. Now the cricket has to know where the wind is coming from, and he has 4 neurons to do it with that are assembled in a Descartesian plane, and every single hair cell is collecting from a certain direction. What this says is that you can reconstruct the original direction of the wind if you have the tuning curves from these neurons. That is, you can decode the world as a function of the encoded percept, the encoded sensory stimulus. And this is, I think, why you find so much constancy and variability in the brain, because you have certain functional invariances that have to be subserved by some sort of constant DSC at a certain level, but there's also a lot of biological type of variability. Some cells in the brain recognize Jennifer Aniston. And you know, there's not that many cells in the brain that you can recognize every single person. So this is it's a Jennifer Aniston invariant cell. So she gets this sort of Jennifer Aniston feature recognition. Modern neural networks also will have this sort of behavior in which they're more active to faces of the person you want to recognize after training. Also so we have distributed, we have distributed representation in our brains. That means we recognize not only the objects that are ecological, but also abstract objects that are these high-level features that can then compose more interesting, more complex scenarios. And they're distributed in the brain. So these are arguments for which convolutional type deep neural networks look very much like what the brain looks like. I'm not going to go into this in detail. And the essence here is pattern recognition. So this is a perceptron, does pattern recognition. I hope that you've heard about perceptrons before, but you know, this is the idea that you can recognize a picture by tuning the receptive fields of contributor neurons, and by creating networks like this, and by using multiple hierarchies. So in this case, we have here a brain hierarchy in which in V1 is your virtual cortex in the bottom. You look into orientation features. In V2, you look into coordinate features. In V4, you look into more complex features, such as objects, faces, and things like that. And these hierarchies, they are very close to what we see in the brain. So convolutional neural networks owe quite a bit to what we have inferred just by looking. The deep networks come originally from ideas from Fukushima. So this is a paper, the neocognitron in, I think, 77, where they were taking this idea from tuning curves, he was Fukushima, and creating a deep network that would reconstruct entire patterns on the basis of small convolutional patterns. And give me another 2 minutes and I'm going to finish. So we're bringing things together. We need not only the physics of it, but also the computation of it. But we need to put them all we need to make it be, right? We need to constrain ourselves from the level of physics, from the level of computation, to the level of the brain. We want to understand how the brain works, and so this is the way that we approach the question, by creating mappings between functional components of the brain with the invariant transformations that we believe they are tuned to perform. This is a model of the brain called SPAWN, and it's made of spiking neurons, single spiking neurons. In Nengo, you might have already heard about Nengo, and ENGO. Is programming language for to create complex cognitive models in spiking neurons, also deep learning models. And it makes very strict assumptions about what the different areas of the brain are doing, but it does it with spiking neurons, and it does it in a way as to try to be compatible with the actual problem. So this is a video from Chris Elias-Smith explaining kinds of things that Nengo can do, and I have only one more slide after this.
My colleagues and I at the University of Waterloo have built a large-scale model of a functioning brain called SPAWN. The model receives its only input.
I hope you can hear my sound.
And controls a simulated arm. In this video, we see both the activity of the various brain areas and decodings of the neural representations in those areas. The first task is a recognition task. SPAWN writes the digit it sees in the image. Individual neuron voltage spikes are shown scrolling through the thought bubble at the back of the brain. The decoded value is displayed on top of that activity. The second task is called copy drawing. Instead of just recognizing the digit, the model must reproduce the style of the digit. Here it successfully copies 2 styles of the number 2. SPAWN's representations are thus useful for both categorization and encoding fine perceptual details.
So what I wanted to get across here is that we in neurocomputation, we're getting closer and closer to computational neuroscience. We're looking into these spiking neurons, and we're trying to see if they actually explain certain features of the activity of the brain. And so we are ultimately interested in the idea of consilience between these seemingly disparate fields, computational neuroscience and neural computation. So neural computation focuses on function, what we can do with neuron. Computational neuroscience focuses on biophysics, the spikes and the activity. And so the assumptions about what is understanding, what is good, differ. And so the explanatory goals differ quite a bit, but we are slowly getting to the point that our main commitment should be to the ultimate explanation. That what will satisfy most of us when we're trying to answer the question, can we explain the brain, or at least the yes, but part of the explanation. So how much of it can we actually get to?